So today we will learn the chapter 24, the final chapter in our this semester. The title is Bacteria, the Archaea, and Virus. Chapter 24 is composed of the 4 subjects, starting from the bacteria and archaea, and also we will learn the prokaryotic diversity, and also finally we will learn the virus. Okay, so this slide we will cover the first subject, bacteria and archaea represent the first split in the tree of the lineage. Okay, so this is the origin of life. Initially, the bacteria and the archaea, they are splitted. At that, eukarya, our eukaryote is also finally splitted, is like this. Okay, so this is eukaryote. We belong to the eukaryote. Okay, so 2 endosymbiotic events starting from the cyanobacterium. So cyanobacterium, they can do photosynthesis, and also proteobiome, they can synthesize the ATP energy. So our eukaryotic cell engulf the cyanobacterium and proteobacterium. After that, it becomes a chloroplast and it becomes mitochondria, is respectively. Okay, so this is a major group of living world composed of the bacteria, archaea, and eukarya. So this is table 24.1. It summarizes characteristics of bacteria, archaea, and eukarya. So bacteria, so they don't have the membrane enclosed nucleus, but bacteria, they have the membrane enclosed organelle. They have few of some of them, they have the membrane enclosed organelle. For example, this is cyanobacteria. So they are filled with tightly packed holes of their outer membrane. It looks like this. Okay, so this is looks like thylakoid. Some of the bacteria have the membrane enclosed organelle, but not many. Only the cyanobacteria. And also archaea, they don't have the membrane enclosed nucleus and membrane enclosed organelle. And the prokaryote, they have the membrane enclosed nucleus and membrane enclosed organelle is like this. You have to know to solve the final term exam. Okay, and also the important thing in this table, for example, peptidoglycan in cell wall, bacteria is present. The archaea, they don't have, eukaryote, they don't have. We will see details in the next slide. Finally, the bacteria, they do have the plasmid, circular DNA, and also the archaea, they also have the circular plasmid is like this. So they have the bacterial DNA, and also archaea, they also have the archaeal DNA. In addition to the DNA, they have a circular DNA. We call it as plasmid. But eukaryote, they nearly don't have the plasmid. Okay, that's why we call it as a layer. It's like this. You have to know. So in this slide, we will compare the bacteria's cell wall and also archaea's cell wall. For in chapter 5, we already covered that prokaryotic cells have the prokaryotic cell is divided to the gram-negative and gram-positive. For example, gram-negative, they have a thin peptidoglycan layer between the out and the inner membrane. How about the gram-positive? They are very thick peptidoglycan layer outside of cytoplasmic membrane. That's why after we did gram staining, they look like the pink. After we did the gram staining, so gram staining cannot, so they look like the purple is like they are very the dark stroke, dark color is like this. However, the Archaea, they don't have the peptidoglycan, but they have pseudo. But actually, this is not peptidoglycan. They have pseudo peptidoglycan, or they have the protein layer. We call it as S layer. Okay, so Archaea they are a little bit different from the bacteria. So they are pseudo peptidoglycan, or protein layer is like this. Now in this slide, we will cover the bacterial cell shape, starting from the sphere and the rod and the spiral is like this. So sphere, we call it as a cocos, okay, cocos, and we also call it a cocci, okay. So sphere is they look like a sphere. An example is enterococcus. So enterococcus, usually in our gut, there are lots of enterococcus bacteria in our gut. Okay. There's another shape is rod shape, like this. Okay, so we call it as bacillus or bacilli. Okay, example is the E. coli. So E. coli looks like the rod bacillus shape is like this. Another example is spiral. We call it as a spirillum or spirilla. Okay, so their structure is like this, and the very famous example is the Leptospira. So they have a spiral structure is like this. Okay, so this is bacterial shape is the sphere or rod or spiral. Okay. So another important thing is the bacteria lateral gene transfer happens in bacteria, only in bacteria. Okay, so DNA or genes moves laterally from one lineage to another. On chapter 21, we already covered what is lateral gene transfer. DNA or gene move laterally from one lineage to another. This is we call it as lateral gene transfer. Okay, so due to the lateral gene transfer can lead to the mismatched gene tree. This is original the tree. However, the lateral gene transfer happens, so it made another gene tree. They are not the same. So mismatched gene tree. So we will see some details. For example, between species C and species D, gene X is laterally transferred from species C to species D. Okay, after that, it made another gene tree. Sequences of gene X reveals the close relationship between C and D. So mismatch is happens. So that's why people use consensus tree. So people made the tree by comparing the multiple gene. So in this case, they compared only one gene, gene X, only one gene. But consensus tree, they are compared blue and the this is the orange and the red gene, multiple gene. Okay, so even though one gene is laterally transferred, they compared multiple gene, so they have their tree is exactly same as the original tree is like this. So consensus tree is used to avoid mismatched gene tree by lateral gene transfer. Okay. 잘 듣고 있나요? 조주영, 너 수업 듣고 있어? 수업 안 듣고 있네, 이 학생. 고선아, 수업 듣고 있어? 아르바이트 하고 있어? 이호정, 수업 듣고 있어? Rhea, I'm marrying. Are you watching the class?
Moi, dis-lui que je peux pas, genre.
Okay, it's okay. 이가연, 수업 듣고
네.
있어?
네네.
너 왜 캠을 안 켜? 나상현, 수업 듣고 있어?
네, 듣고 있습니다.
왜 캠을 안 켜?
캠이 없어서.
이규빈,
네.
수업 듣고 있는 거야?
네.
캠을 왜 안 켜?
컴퓨터라서 캠이 안 켜져.
오케이. 하은이 수업 듣고 있는 거야? 하은이 수업 듣고 있는 거야? 수업 듣고 수업 잘 들어 알겠지? 평소처럼 수업 듣지 않는 수업을 하고 있는데 PDF도 안 켜놓고 반성하고 알겠지? 오케이. Okay, so okay. Consensus tree once again is constructed by comparing the multiple gene, okay? Okay, now we will go to the prokaryotic diversity. Okay, so this is bacteria. Okay, so they have the Heidobacteria and the hyperthermophilic bacteria. This species is very rare. So today we will not cover these two species, and we will start from the Formicutes. Formicutes Korean name is Hubyokgun. Korean name is also very difficult to understand. Okay, let me start. Formicutes. So they are low GC gram-positive. They are gram-positive bacteria. Okay, but they have a GC value is very low. The GC value is very low compared to the 80. Okay, this is recorded as Formicutes. Okay, and some of them they have the endospore. So this is their host cell, and this is they made the endospore. So they have the heat resistant resting structure with the top, very top, they have top the cell wall. So even though in higher temperature, they can survive. promiscuous, they made the endospore like this. So even though in the high temperature, they can survive. Okay. The very famous example is Staphylococci. So this is Staphylococci. They look like the grape cluster arrangement, grape. So it can cause skin infection, or the wound infection is like this. Okay. So usually all of the bacteria, we can kill it by heating the temperature. However, especially the Staphylococci, and also they can make the endospore. Even though we increase the heat, we cannot kill the Staphylococci is like this. Okay. Another one is the next one is the actinobacteria. So actinobacteria in Korean we call it as 방선균. So they have the previous promiscuous, they have low GC gram positive, but this one is the GC, they have value is high compared to the 80. This one we call it as actinobacteria. And also they have several branch like this. They forms branch filament. Okay. The very famous example is Mycobacterium tuberculosis. So it causes the tuberculosis. So we call it as they have the persistent cough, and whenever they did cough, they have blood in the cough. So this is tuberculosis symptom. Okay, so actinobacteria, okay, they made the main cause of the tuberculosis. It's like this. Okay. Next is cyanobacteria. This is cyanobacteria. So they are gram negative. So from cyanobacteria, they are gram negative. Previously, we learned that the cyanobacteria, they make oxygen through photosynthesis. So they receive the light, and they also use carbon dioxide. So finally, they made the oxygen. So gram cyanobacteria, they make this kind of filamentous structure, or they show that they look like the spherical balls. Cluster of spherical balls is like this. This is their structure is like this. Cyanobacteria. Okay. Next one is the Spiroheta. Spiroheta is near, in Korean we call it as Spiroheta. So this one is also gram negative. Okay, so they have several endoplasella structure. Okay, so they have the several endoplasella structure like this. Okay, so each endoplasella can rotate it. So another endoplasella can rotate it. So they make the screw-like, corkscrew-like movement. So this is corkscrew. This is corkscrew-like movement is like this. So the human parasite and pathogen belong to the Spiroheta. So very famous example is Syphilis. So Syphilis, so one of the Spiroheta, make the syphilis. Syphilis in Korean, we call it as medok. So this is the primary syphilis is occurs in the man's sexual organ. So this is syphilis, and also secondary, tertiary syphilis is happen like this. Okay, so spiral heter, they make the corkscrew-like movement, and also they are pathogen. They cause syphilis. Okay, sexually transmitted disease. Okay, syphilis. Okay, another one is the chlamydia. Chlamydia is they are very small, and also they are gram-negative. Okay, so chlamydia, they live only as parasite. They cannot survive itself, but they live only as parasite in cells or other organs. So they have two forms: elementary body, we call it as EB. Another one is they have the reticulate bodies. Okay, their structure looks like like net, like this. They have this kind of reticulate structure like this. Okay, in Korean we call it as a 망상체, reticulate body, RB. So initially the EB, so host cell is uptake to EB. So EB is converted to RB. So their structure looks like this. Okay, and also RB is replicated. Finally, RB is converted to EB. Finally, the EB can be released and act like this. So their life cycle is elementary EB, and the RB is like this. Okay. Final one is in bacteria, proteobacteria. Okay, this is the largest group of bacteria. Most of bacteria belong to the proteobacteria. For example, E. coli, we already know that. Also several another Vibrio, it causes the cholera. They also belong to the proteobacteria. Okay. So especially the Agrobacterium tumefaciens. Okay, this is also belong to the proteobacteria. So they cause crown gall in plants. So they make the cancer in the plants. Okay, crown gall in plants like this. Okay, so today we cover all of species of bacteria. So we did not cover HADOR, hyperthermophilic bacteria. So we started from the Promecutis, Achino, Cyano, Spiroheter, Chlamydia. Finally, we cover proteobacteria. Okay. Now we will go to the Archaea. Okay. So Archaea, in Korean we call it as a 고세균. Okay, Archaea, they lack peptidoglycan layer in cell walls. Archaea, they have pseudo peptidoglycan layer, and they have the protein layer is like this. So and also they are composed of the lipid bilayer like this, but membrane lipid they have a fatty acid. They are linked to the glycerol, but they are linked by either linkage. Okay, oxygen and carbon, but carbon has hydrogen like this. This linkage we call it as the ether linkage. Previously, we learned that, so this is the ether linkage like this. Previously, we learned that lipid bilayer, so lipid is the linked by ester linkage. So they have the 3 fatty acid, the 1 glycerol. So they combine to each other, combine to each other, combine to each other. This one is we call it as ester linkage. So oxygen combined to the carbon, carbon as oxygen like this. This linkage, we call it as ester linkage. However, Archaea, their linkage is the ether linkage. In Korean, we call it as heterokerap, ether linkage. Very important. This is very important. Okay, you have to know to solve the final term exam. Okay. Okay. And also, Archaea also has a lipid bilayer like this, lipid bilayer. Also, they have the lipid monolayer. Both of them is found in the Archaea. For example, they have 1 fatty acid. They have 2 head is like this. Okay. So Archaea lipid bilayer and the lipid monolayer are both found in the Archaea. And usually the Archaea lives in the extreme environment, especially the high salinity, high temperature, or high or low pH. Okay, Archaea is there, lives in extreme environment. Okay, let me go to here. So we covered the bacteria. So now we covered Archaea. There are several kinds of Archaea. The most important thing is that Krenakiot. In Korean, we call it as Krengogyun. Krenakiot. Okay, so this is belong to here. So they are thermophile. Hot spring, the temperature is nearly 70 to 75 centigrade. Okay, here, even in hot spring, some organism survive. That one is the Krenakiotis. We call it as Krengogyun. They are belong to the Archaea. Okay, they live in hot spring. And another one is acidophile. They can live in pH 2 to 3, but their internal pH they can maintain 5.5 to 7. Okay, so this is the acidophile, or the thermophile is belong to the Krenakiotis. In Korean, we call it as Krengogyun. Okay, another one is this one. So Ureakiotis. In Korean, we call it as a Uregogyun. Okay, they are extreme halophiles. So we call it as halophile, non-halophiles, and slight halophile, moderate halophile, extreme halophile. So they can live it in the higher concentration of salt, because they are extreme halophile. They require higher concentration of salt to survive. Okay, so Ureakiotis, in Korean we call it as a Uregogyun. They are extreme halophiles. Okay. Okay, so Archaea is composed of the Krenokiotis and Ureakiotis, and the other 3 is there, but they are very rare. So we covered the major species, Crenachiotis and the Eurychiotis. Okay. Now we will cover the third subject. Ecological communities depends on prokaryote. So usually the prokaryote lives in community. Okay, so this is prokaryote. So, prokaryote is bind to the surface. First of all, they did the reversible adhesion. So it means that they can attach or adhere to surface, but they can detach it. Okay, so this is we call it as a reversible adhesion. But times go on, they did irreversible adhesion. They attach it to the space, irreversible. After that, they make the microcolony. After that, they make the microphyllum. Okay, so they coated the space. Okay, so in this case, antibiotics cannot penetrate the biofilm. So it means that it's difficult to kill the bacteria in this biofilm. So usually the prokaryote, to protect themselves, they make the biofilm is like this. Okay, because after they make the biofilm, antibiotics cannot penetrate them, so they can survive. Okay, this is the why prokaryote they made the community. For example, antibiotics, they can kill this one, 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 but they cannot kill after they make the biofilm like this. They cannot kill this one. Okay. No, and also the prokaryote, okay, we call it as a microbiome. So we call it microbiome. So body is micro. In our body, we have lots of the prokaryote. In our mouth, in our this one, and also in our gut, we have lots of the prokaryote microbiome. So usually the human health depends on the health of microbiome that lives in our body. For example, in our gut, we have lots of the good microbiome and bad microbiome. So the microbiome can control the diabetes or obesity. So if we have a harm microbiome in our gut, we will become the obesity, or we will have diabetes. Is like this. Okay, and also humans, female, they have the Virginia. Okay, Virginia also have lots of their microbiome is there. So this is the feminine cleanser. So women use feminine cleanser, but it can kill all of the microbiome. So Virginia, they have the very good microbiome or bad microbiome. However, the feminine cleanser, they can kill all of the microbiome. So some people, so doctor said that feminine cleanser is not good for the woman health because they can kill both of them. Okay. And also prokaryote they belong to the aerobal or unaerobal. The prokaryote organism that requires oxygen for growth, we call it as aerobal. They use this oxygen. However, unaerobal, they do not require the oxygen for growth. And also there is the obligate on the aerober. This is the, in this aerober, oxygen is poisonous to them, but most of them is facultative anaerober. So they shift between the aerober and anaerobic mode. If they have the oxygen, they become the aerober. If they don't have the oxygen, they will become anaerober. Most of the anaerober belong to the facultative anaerober. They can shift between aerober and anaerober. Okay, or prokaryote, they can be belong to the photoautographs or photoheterotrophs. Photoautotrophs or photoheterotrophs. Okay, both of them use the light, solar light. Photoheterotrophs also use the solar light as energy source, But photoautotrophs uses carbon dioxide as a carbon source. However, photoheterotrophs use the organic compounds from other organisms as a carbon source. So finally, they will make the glucose, and this is glucose. They will make the glucose is like this. So we can divide it by two photoautotrophs or photoheterotrophs. It's like this. Another one is the chemoautotrophs, chemoheterotrophs. Let me start from the chemoautotrophs. So chemoautotrophs get energy. They make the ATP by oxidizing inorganic compounds. For example, they oxidize the ammonium. After that, they make the ATP. However, and also autotrophs, they use the carbon dioxide as carbon source. However, chemoheterotrophs, they use the get energy by oxidizing the organic compounds, glucose as carbon, so we call it as organic compounds, by oxidizing organic compounds. Also, they use the organic compounds, so in this case, they also use glucose as a carbon source. So this is, we call it as a chemoheterotrophs. Okay, you have to know what is chemoautotrophs, what is a chemoheterotrophs. You have to compare to each other, or you have to compare to photoautotrophs, what is photoheterotrophs, and what is aerober and what is anaerober. Okay, important. Okay. So here we summarize table 12.2 summarize how organisms obtain their energy and carbon. For example, photoautotrophs, they use the energy source as light, carbon source is carbon dioxide. Photoheterotrophs, they use energy source as light, but their carbon source is the organic compounds. Chemoautotrophs, they use the inorganic. Inorganic means that they don't have the carbon. So in this case, they use the nitrogen, ammonium as the ammonia. After they oxidize the ammonium, and they made the energy. Okay. And also they have the chemoheterotrophs, is like this. The energy source, they use the organic compounds, and also the carbon source, they also use organic compounds, is like this. So now we will cover the final subject in the virus. Okay, virus have a multiple evolutionary origin. Okay. 수업 잘 듣고 있나요? 강민정 수업 듣고 있는 거야? 사라져 있네. 하은이 수업 잘 듣고 있는 거야? 조은솔,
네.
조은솔 수업 잘 듣고 있는 거야? 조은솔, 오케이. 조은솔, 평소 수업 시간대로 하면 안 돼. 수업 잘 들어야 돼. 오케이. 우성아. So, as I mentioned earlier, so the most important thing in life is patience and persistence. And also, most important thing is in our important thing is don't be late. Okay, I said several times, and also several times, don't be late. Well, I found that several students still be late. Okay. Okay. 이가연,
네.
너 왜 안 내? 안 냈어. 왜 안 냈어?
제출하는 걸 까먹었어요.
특히 뭐 할 말이 없습니다. Okay. So I will give you the final chance. Okay. Okay. Submit homework. Okay. I will give you another final chance. Okay. 손세안. 손세안. I will give you the final chance. Okay.
Yep.
Submit homework. Okay.
Yep.
Final chance. Okay. 김하은. Final chance. Okay.
네.
Okay. And also, so after finish this semester, you will have the summer vacation. What is the most important? What you have to do during summer vacation? 윤지원, what you have to do? What is your plan? 윤지원, what is your plan during the summer vacation? 조은솔, 너의 플랜은 뭐야?
네.
너의 플랜은 뭐야? 정효주, what is your summer vacation plan?
저 시험 공부요.
조주영, 수업 듣지도 않고 있지? Okay. So this lecture was the English lecture, based lecture. Some of them can understand the lecture, but some of them did not understand the lecture. Most important thing is during the summer vacation, you have to do English, studying English. English is most important to apply your job or to apply the graduate school. 그러니까 영어가 안 되면 안 됩니다. 알겠죠? 윤지원,
네.
너 졸업하고 뭐 하고 싶어?
저 취업이요.
취업, 어떤 데 취업하고 싶지?
그것까지는 아직 잘 모르겠습니다.
윤지원, 내가 100% 확신으로 이야기해 주는데, 너 너 한 가지만 하면 취업 완벽하게 되거든?
네.
가르쳐 줄게, 이제.
네.
토익을 한 950 넘으면 취업할 수 있습니다. 100% 확신입니다. 토익을 고득점 받으면 한국에 있는 대기업뿐만 아니라 모든 회사 다 갈 수 있습니다. QC, QA가 되려고 해도 토익만 950 넘으면 무조건 다 될 수 있어요. 이거는 변하지 않는 룰입니다. 회사에 있을 때 사람들 뽑을 때 서류 전형에서 그럴 때 토익으로 다 걸러요. 학교로 그러는 게 아니고 토익으로 다 걸러요. 서류 전형에서. 토익이 높지 않으면 서류 전형을 통과할 수가 없어요. 그래서 영어가 중요하다는 거예요. 양소원, 너 렉처 하는 거 들려?
네.
그 영어 다 들렸어?
아니요.
그 방학 때 노력해야 되겠지, 맞지?
네.
오케이? 그러니까 100% 슈어합니다. 토익만 950 넘으면 못 갈 데가 없습니다. 저희 졸업생들 봐보면 토익이 950 넘은 애들 다 취업됐어요. 학부 마치고도 SK 바이오사이언스뿐만 아니라 삼성 바이오로직스도 많이 가고요. 에피스도 가고요. 셀트리온도 가고요. 그런 학생들은. Okay? Okay. So during summer vacation, you have to study very hard, okay? And Rhea, Marin,
Yeah.
when do you come back to France?
Probably the end of June for Marin and a bit later for me.
Oh, okay. So maybe you can, there are after finish final term exam, maybe there are several places to visit in Korea. There are very several, so you can visit there, okay?
Yeah, we already plan to go visit some stuff.
Okay. And good luck in your future, okay, Leia and Mari.
Thank you.
Okay.
Thank you very much.
Okay. So now we will cover the final subject. Virus. So virus have a multiple evolutionary origin. So usually we divide it two species. One is RNA virus, the other is DNA virus. Okay. So for example, RNA virus, there are three kinds of RNA virus. One is negative sense single-strand RNA virus. Another is positive sense single-strand RNA virus. COVID-19 virus is belong to the positive sense single-stranded RNA virus. And also we have the RNA retrovirus. HIV is belong to the RNA retrovirus. But today I will not cover in details. So if you are interested in virus, in our department, Professor Yejongyong, he opened virus subject. So today we will cover only 2 slides, but in the Professor Ye's lecture, so during one semester, he covered what is virus. But today we'll cover that virus we are divided to RNA and the DNA. DNA is the double-stranded DNA virus, and also double-stranded Mimi virus is there. But today I will not cover in details. So usually the x-axis is genome size and the y-axis is mutations rate. So usually the viral genome is very tiny, but they have the higher mutation rate. Okay, that's why COVID-19, they mutated very frequently. So it is very difficult to make the vaccine because they mutated very frequently. And also viral genomes can evolve multiple times from different cellular species. Okay, for example, one COVID-19 impacted me, for example, one case, and also that virus can impact other people, and also other people, and other people. At that, the final virus have a different genome. Because viral genome can evolve multiple times from different cellular species. That's why it is very difficult to make the virus vaccine. Okay, so this is virus, the characteristics. Viral genome is very tiny, but they are higher mutation rate. Okay. Okay, so today we cover chapter 24. So we finished all of our, so we plan to the cover from chapter 1 to chapter 24. We covered everything. Okay, so do you have any other question today's lecture, or do you have any other question in the final term exam? Okay, good luck. Good luck in your final term exam and good luck in your future. 근혁성, 박수빈, good luck. 강유빈, 강지은, different department.
감사합니다.
Okay, good luck. Okay, today we will finish our class. Okay, done. Okay.
저 혹시 늦게